Welcome to Capital Considerations, the podcast that takes complex ideas from the investment world and makes them accessible to everyone. I'm your host, Tony Roth, Chief Investment Officer of Wilmington Trust.
Today we start the first episode of a two-part examination of the deep science behind the coronavirus. In this episode, we'll be joined by microbiologist Susan Weiss from the University of Pennsylvania, Perelman School of Medicine, where her lab focuses on coronavirus pathogenesis. She is also a co-director of Penn's newly founded Center for Research on Coronaviruses and Other Emerging Pathogens. Dr. Weiss is here to discuss the biology and epidemiology of the coronavirus and how the scientific community is working to better understand the intricacies of this virus. Thank you, Dr. Weiss, for joining us today.
Thank you. It's very nice to be here.
Well, it's great to have you, and we're excited for the conversation. And we've had a lot of conversations with others around the various medicines that many people around the world are working on, whether it be therapies or vaccines. And it occurred to us that we really need to do a primer, if you will, a biology 101 on viruses for our audience, since viruses are very unique and tricky creatures. And we thought it would be really helpful to start with some of the foundational elements and understanding what a virus is and how it works.
Well, a virus is really a very simple thing that's very clever and can do all kinds of things to its host cell. But the virus itself is actually, the particle is called a virion, and it's composed of a piece of genetic material that can either be DNA, which you've probably heard of, which is our genetic material, or it can have only RNA as its genome. And this RNA or DNA is wrapped up with proteins in kind of a capsid, it's called. And then many viruses, coronaviruses for one, have a membrane around them. And that membrane is made out of lipids, which are really just fats. And that membrane comes from the host cell. And in that membrane, it has other proteins sticking out of it. And these proteins that stick out, are in the case of coronaviruses, are called spikes. But many viruses have very similar proteins. And that's the protein that attaches to the host cell. It recognizes a receptor on the host cell. And through this interaction, the virus enters the cell. One thing that's important to note about viruses are they're not really alive. They're able to replicate, but only inside of the cell. If you have a virus just sitting on a tabletop or in water or whatever, it can't really replicate. It's not really dead or alive. It's different from a bacteria.
But if we touch a surface that's been where a virus has been recently deposited, we can contract the disease. So when you say it's not alive, in what state is it? If I
thought you were going to ask that. So it's kind of, I would guess it's like, it's static. It's not growing and it's not dead. It's sitting there. And it won't last there that long. I mean, as a period of time, it'll decay. All viruses will have a half-life. They can be denatured or fall apart from heat or from even UV light or anything like that. So yes, if you put down some virus on a tabletop and someone else comes by and touches it and puts it in their nose or eyes, yes, they can catch the virus, but it's not going to actually replicate. The amount that you put down on the table is just going to decrease over time rather than replicate.
So the spike somehow enables it to gain entry into the cell in the case of a coronavirus, which has the spike. And then what happens next?
Okay, and that's a really general thing. So like a herpes virus has other proteins. They're not called spikes, but they have the same function. So many viruses have the same kind of function. And so that protein, it recognizes a receptor on the host cell. That could be, they're all different proteins for different viruses, and they have other functions, these receptors. They just happen to be proteins that the virus can kind of latch onto. Once it latches onto it, the spike protein undergoes a change in its conformation, and the virus, as I said, has a fatty lipid envelope, it's called, around it, or a membrane. That membrane fuses with the host cell membrane, and then the inside of the virus or the capsid can get kind of pushed or released into the cell, and so that it can start replicating. So it leaves the membrane behind, and just the inner part, or the capsid, gets into the cell and begins the process of replicating.
And so within the host cell, the virus is actually just growing in the sense that it's just replicating itself over and over again?
Yeah, you wouldn't call it growing because it doesn't get bigger and it replicates. It kind of takes over the host cell. So it leaves its membrane. We call it uncoating. So now the DNA or the RNA and its proteins surrounding it are, we say, it's sort of released into the cell. And once it's there, it starts making proteins, and then those proteins can start replicating the RNA. So it's not like you get, like bacteria, they divide and you get 1 to 2 to 4 to 8. With viruses, they just start spinning off huge amounts, many numbers of thousands of copies of that RNA, of its genome RNA. It replicates itself and it makes messenger RNAs. Those are RNAs that encode for proteins. So it makes lots of RNA and lots of proteins. And then it reassembles itself into new virus particles that then leave the cell. Sometimes they kill the cell, sometimes they don't.
So what does the disease consist in? In other words, we've got this virus that's fused into the cell, and it's replicating itself. So I could understand that if it kills the cell, that would seem, I'm not sure what the right word is, a pathogenic activity. Is that what the coronavirus is doing? It's actually killing the cell, or is the disease consist in something else?
Well, it's sort of a combination. It may kill some cells and not kill other cells, but in a way, the virus doesn't want to kill everything, because if it killed everything, it couldn't replicate. It wants to make more of itself and then go on to the next cell and replicate in the next cell and the next cell. But one of the side effects of infection is the host cell is going to respond to that. So the host cell is going to make lots of, like cytokines. You've heard of, probably everyone's heard of the cytokine storm. So that cytokine storm is initiated by the cell realizing that it's infected. It responds by those cytokines are meant to get rid of the virus, but an overexuberance can be destructive to the lungs in the case of the coronaviruses, but just can be destructive to the host.
Okay, so that's the basic life cycle of a virus. That's our basic foundation.
Yep.
Okay, so in the case of the coronavirus, is there anything, and you've studied coronaviruses for decades.
Yes.
What makes this coronavirus fundamentally different than other coronaviruses, or is it?
It's true. It's not fundamentally different. If we look at the genome, which is again the genetic material, and compare that to SARS coronavirus, the original SARS coronavirus, I call sometimes SARS-1, from the one that was emerged in China in 2002, they're very, very similar. I think 95 or 96% of their sequences are similar. So it's not like you look at it and say, wow, this one has a new gene that maybe is making it more pathogenic. It's pretty tricky, really, because all coronaviruses have pretty similar genetic materials, or I shouldn't say all of them, but these lethal ones. But they're behaving so differently, and to me, that's really mysterious. What you said is absolutely correct. This virus seems to be more transmissible. It seems to be transmissible before someone actually shows symptoms of being sick. And it seems actually the lethality is much lower than the original SARS or MERS coronaviruses, just 1% of infected people. But it's so tricky because we didn't see with those first two epidemics, there weren't people walking around on the street with the virus that were not apparently sick. So, and like I said, it really is puzzling that we don't know why that is from the genetics of the virus. Not at all obvious.
And is it puzzling at all that there's such a wide spectrum of individual responses to it? I mean, obviously, you're always going to have people that have compromised immune systems, or potentially people that are more elderly, I would imagine, are always more susceptible. But even within the range of individuals that we see that tragically are felled by this virus, you see people that are seemingly young and healthy in certain cases, or even if they're not ultimately killed by it, they get very, very ill. Then there are other people that, we hear the 70 or 80% of the people that contracted have no symptoms whatsoever. Is that typical, or is that a bit unusual compared to the other coronaviruses?
Compared to the other coronaviruses, I think it's very unusual. That people, like I said, people that had SARS were very sick, and something like 10% of them died, much more than we're seeing with this coronavirus. But because people were so sick, they were put in the hospital, they were isolated, and it was easy to contain the virus, or relatively easy. It lasted for 8 months, and then it was gone. And I don't think anybody thinks that's going to happen with this one. And that was also true with MERS coronavirus. MERS coronavirus is still infecting people in the Middle East, but again, people are really sick. And I think here the issue is that we just can't tell when. We just can't tell who has the virus. And I asked a friend of mine who's a medical doctor, I'm not a physician, does this seem really surprising? Because to me it did. And he said, well, for example, other viruses like some of the flaviviruses, West Nile virus or Zika virus, you can have very inapparent infections, and then every now and then somebody gets really sick. So maybe it's not that unusual. It is unusual for a coronavirus. And one of the reasons...
Such a varied impact.
I think so. And one of the reasons that it's so dramatically that people are getting so sick is that we have no immunity to SARS coronavirus. We've never seen it in the U.S. before. And even I don't know if people that had the original SARS, if they would be immune or semi-immune, if they were sort of immunized against this future SARS virus by having the original one, I don't really know. But it's sort of like when flu comes around, most people have had some flu or a flu shot, so even if they get infected, they may not get that sick. Whereas here, we're completely naive to it, so we're like sitting ducks, I think.
Right.
Yeah.
That's interesting, the idea that there could be some immunity from another virus that may be very similar but not exactly identical to this virus, a different kind of coronavirus, but very few people would have even had exposure to those earlier coronaviruses.
But it would have to be relatively close. I said, for example, SARS coronavirus. Like if there were a vaccine, had been a vaccine, maybe that would have worked against this virus. I think for MERS coronavirus is going to be too far. I don't think it would give any kind of immunity to this virus.
So we have some basic stages of the life cycle we've just defined, which is, at least in terms of its relationship to its ultimate host, it's the entry, and then there's the replication, and then it moves on to the next cell. So when you think about those stages, are there particular stages from an intervention standpoint that medicines try to target to try to stop the activity? And are there certain stages that tend to be more successful than others?
Well, we don't know what's successful yet. Everyone's trying to figure that out. But I mean, we know from other viruses. So the first step that's often targeted is viral entry. I talked about how the spike protein attaches to its receptor, and then it mediates what we call fusion, which is fusion of the viral membrane envelope with the cell envelope and release of the capsid into the cytoplasm of the cell or into the interior of the cell. And for coronaviruses, there are two possible pathways they can use to enter. This may get a little detailed, but I'll try to explain it simply.
No, that's okay.
The spike attaches to the ACE2, which is the angiotensin converting enzyme or the receptor, and it can either at that point just fuse right into the cell, right at the membrane there, or it can be endocytosed into the membrane. That means that a vesicle kind of envelops it into itself, and it's deposited into the cell in a vesicle, like in a bubble kind of thing. And then from that vesicle, it fuses into the cytoplasm. And that may seem like kind of a subtle difference, but the reason why that's important is that the second method through that bubble requires a low pH or an acidic pH, and there's some enzymes in that vesicle called catepsin that has to be activated to release the virus particle, and that only happens at low pH. Okay, so chloroquine is one of the drugs. And hydrochloroquine are drugs that have been bandied about and probably in clinical trials now as an antiviral drug. And the way that chloroquine works is it prevents that acidification of the endosome. So it's a kind of general mechanism. So any virus that requires that low pH step should be sensitive to chloroquine, or we'll find out if it is.
Do we know that the coronavirus gains entry or fusion through that second method?
It's a little more complicated. So coronaviruses can use both pathways. The first pathway doesn't require the low pH, but it may require an extra protease step. So the spike protein has to be cut or cleaved in two places to activate it. this gets a little bit complicated too. And those cleavages depend on sequences in the spike protein, and they also depend on enzymes on the cell that makes the virus and the cell that the virus is entering. So it's a kind of a complicated mixture of what the sequence of the spike is and what enzymes are available to cut the virus. So some coronaviruses will enter mostly through the cell surface, some will enter mostly through the endosome and need the low pH step, and some can do both. And on top of that, it's even more complicated because different cell types express different enzymes. So this virus may be more likely to use one pathway or the other in one cell type or the other. Does that kind of make sense?
But essentially what you're going to tell me, I think, unfortunately, is that we don't know yet which COVID-19 is in terms of how it gains entry, right? Well,
we know a little. There's some data about that. There's some studies that say that this enzyme called TMPRSS is important for entry of SARS-CoV-2, and that may be only on a particular cell type that it was tested on. So that if the virus uses TMPRSS, it would suggest that it's going to go in through the membrane directly and not through the endosome. But if it were to do that, then chloroquine really shouldn't work. So to me, if you use chloroquine, you might kind of force it to go in through the membrane. If you use the TMPRSS inhibitor, you might kind of force it to go in through the endosome, because maybe it can go in by both pathways. Some viruses can go in through both pathways. So my prediction is that probably and this is just a prediction, that you probably have to shut down both pathways if you want to really shut down viral entry.
Well, so there's been, as I think we all know, I think at best we could say mixed results with the chloroquine. Certainly has been far from a slam dunk. But that doesn't mean that perhaps if it was used in conjunction with one of those other drugs that blocks the other path, maybe the two of them together could be effective.
I would think theoretically, yes. I don't know I don't know if we know exactly that chloroquine only acts by disallowing low pH. Maybe it does other things as well. I've heard that it may have other mechanisms, but I don't really know. But yeah, I would think that if you shut down both pathways really well, that would be like a combination therapy to prevent viral entry.
Are there any other medicines or compounds, if you will, that are either being used off-label or in development that you've heard anything about in terms of being effective at this stage? Yeah, for entry for COVID-19. I don't know whether it be, I think the remdesivir we're going to talk about, that's more of one that targets replication, right?
There are inhibitors of TMPRSS, but that's theoretically one way to go to try to inhibit that. There's also furin is another one of the enzymes that actually it's an intracell. It's inside of the cell, but it also cleaves that spike protein to get it sort of semi-activated for entry. So if you treated cells with a furin inhibitor, this is just all theoretical. I don't know whether drugs like that exist or that they work or anything, but if you could inhibit that furin stage, you might end up producing virus that was unable to infect the next cell. So I think that these host cell proteases are probably a good potential target.
You and your team have spent so many years looking at coronaviruses and evaluating or defining these processes and mechanisms. I would imagine that the work that's going on now to try to identify inhibitors, if you will, antivirals, inhibitors for these activities, it has to stand on the shoulders of all the work that's already been done over the years, so that had we not had MERS, had we not had SARS, we wouldn't be as far along in hopefully getting closer to a solution, whether it's a generic solution that works against all the coronaviruses or something even just specific against COVID-19. All the work that's already occurred has got to be very important in moving us along.
Yeah, I just should correct you that COVID-19 is the disease. The virus is actually called SARS-CoV-2. And the reason it's called SARS-CoV-2 is it's almost the same as the original SARS. So just to be more precise about the nomenclature.
The original SARS was SARS-CoV-1?
Well, it was just called SARS-CoV because we didn't know there's going to be another one, right?
Got it. Okay, of course. SARS-CoV.
Severe acute respiratory syndrome, right? And then MERS is Middle East respiratory, right? So now this one, there was a lot of controversy about the name, but it was named SARS-CoV-2 because genetically it's pretty close to SARS-CoV-1. SARS-CoV.
So all that work that you've done and others around the world is very critical as a building block upon which people are now putting many times more energy into trying to develop these inhibitors or antivirals for the processes that you've just described.
Well, can I go back a little further? I'm not to SARS. So SARS was 2002, but I'm going back to the 60s and 70s. We knew about human cold coronaviruses, OC43 and 2290. People worked on them. They didn't work on them super hard because they were cold viruses and they weren't of that much concern. But there are a lot of animal coronaviruses that were worked on quite a lot, like IBV chicken virus, BCV cow virus, and some porcine viruses, and we worked on mouse hepatitis virus because it was just a model. So it was all those work on those viruses before we even knew about SARS that really built up. That's how we know about viral proteases. That's how we know that they make this long polyprotein and 16 non-structural proteins. A lot of that was known before SARS, so that when SARS arose in China, I can tell you everyone in my field was absolutely amazed. We couldn't believe it. We were shocked because there had been no evidence up until then that coronaviruses could cause severe diseases in humans, which is kind of strange in a way because we knew they could cause severe diseases in other animals. But it was because of all that knowledge. I mean, when SARS arose, it was sequenced. The genome was sequenced really quickly. It was cloned so that people could work with it. It was an amazing amount of work that went on before that. And then when SARS arose, many people started working on these viruses in a way that it was shocking, actually. The field was very small before SARS, and then it grew enormously at that time. And we learned a lot more. The details, like these enzymes I'm talking to you about, people had actually figured out the crystal structures of them. And that's important because if you know the structure, you can kind of model what kind of inhibitor might combine with it and prevent it from having its activity.
So what is the relationship that you and your team play as what I'm thinking in my mind is extremely basic fundamental science vis-a-vis the specialists that are trying to develop these antivirals? Do you interact with them? And are the different drug companies asking you questions about the behavior of this virus? How is the collaboration work across these different specialties?
In the time before 2002, I think the field was pretty much ignored. After SARS, I gave more seminars than I've ever given in my life, and I did talk to some drug companies about, just wanting advice and stuff like that. So there was a burst there. I personally am a really, really basic science. Some of my colleagues are doing more actually drugs, not really drug studies, but looking for inhibitors in their own labs, but on a kind of a smaller level. My colleague Sarah Cherry now at Penn is doing a massive drug screening against this virus. So, if she finds something, I assume a drug company would pick up and want to develop it. But again, we defined what some of these proteins are initially, not just our field, and that's incredibly important. Remdesivir, the reason that people even tried it on coronaviruses is it was originally designed or used for Ebola. And the reason why you might even think it would work on this virus is that RNA, it turns out that RNA polymerases, that's the enzyme that copies RNA into RNA, they're kind of structurally similar even among different viruses. Coronaviruses all have these 16 proteins they make that are conserved. Some of these sort of protein domains, you might find similar ones in other viruses.
So when you put it all together and you think about from what you've seen, and I know that your vision is not comprehensive, nobody's is.
No, it's not.
Is there a particular class of antivirals or a couple drugs or anything that you think at this very early stage seems to be most promising?
I can't really assess that. I can really talk more theoretically. I mean, to me, that I don't know about remdesivir, but a drug like that seems like a pretty good starting point. I mean, these viruses don't change that much anyway. The proteases, I mean, if you inhibit the protease, you really have to stop the infection. So I don't really have a prejudice as to which one might be better than the other. It really just depends how well the inhibitor works. because all of these functions are required for replication.
What's the difference between, changing gears a bit here, the difference between preventing the virus from entering the cell in the first place and what I would think a vaccine is, which would be something that would somehow prevent the virus from getting into my cells? There's obviously a difference between a medicine that's aimed at preventing that initial stage of entry, like chloroquine that we talked about, versus vaccine. How do you compare those?
Well, a vaccine obviously is prophylactic. A vaccine should really theoretically prevent viral entry too, because if you make antibodies that are so-called, you've heard of maybe neutralizing antibodies, that means that they'll neutralize the virus and prevent it. They usually are directed against the spike protein, so if they're really good neutralizing antibodies, they should prevent that infection. So, I mean, in a sense, it's kind of targeting the same step, but if it leaks through and it does if the virus does cause some kind of infection, because nothing's 100%, the new virus will still have to deal with the antibody response from the vaccine. So maybe in a sense, it's looking at both entry and then spread as well. But so is an entry inhibitor.
In the case of a therapy, it's the actual medicine that's preventing changing something in the human body to prevent the entry. But in the case of a vaccine, it's actually stimulating the body to create the antibodies or whatnot to prevent the entry.
You could. So it's a different mediator of the block and entry, but it's still both kind of the goal is to prevent the virus from getting into the cell. The vaccine also will work on any virus that does leak through the cell. If you have a T cell response as well as an antibody response, you might also kill infected cells. So that's an added step that you wouldn't have with a drug, with a vaccine. It depends if the vaccine is inducing a good B cell and/or T cell response.
If you think about the groundswell of interest after SARS relative to previous, where there's many, many more people focused and interested on this, even compared to that, the number of people that are focused on it now is probably an order of magnitude of many, many times.
Or 2 orders of magnitude, maybe.
Right?
Yeah.
So given that massive interest now, of course, the world is trying to solve this, how do you see your work changing or progressing in a different way than it might have had this not happened?
Well, we have a new virus to work on. We want to know why this virus is different. So one of the things, like I said, I'm still doing very basic research because that's where my strength is, I think, and that's where I can be helpful. So we study how the viruses antagonize the host innate immune response. We didn't really talk about the innate response very much. That's interferon, and that's a really early response, not really specific to that virus, but any kind of, when a cell sees double-stranded RNA, which is a product of virus replication, that's seen as a danger signal by cells, and they see that and they make interferon, and they also activate some other antiviral pathways, antiviral activities. And coronaviruses are really good at stopping that or antagonizing that innate response. So what my lab's been interested in the last 10 years or so is like, how do viruses do that? We've worked on that with the MERS coronavirus and the mouse virus. And so we want to do that with SARS-CoV-2, and we also want to create a mouse model. So in order to infect mice with this virus, you have to make transgenic mice that express the SARS-CoV-2 receptor, or ACE2, human ACE2. And so once you have that, we can actually make mutant viruses and try to determine which parts of the viral genome are really causing pathogenesis. So in that sense, we get towards the disease process. And if we uncover things that the virus does to the cell, it will help us figure out what steps to target. Like you can also target host cell pathways and boost them up rather than just trying to, inhibit the virus. So that's where my interest is. Like, how does this virus prevent the host cell response?
Interesting. So let me try to summarize quickly what I think we've covered today. And I'm going to talk about 3 key takeaways. And the third one, I'm going to add a little bit, and you have to tell me if I've got it right.
Okay.
So I think the first one The first two really are around providing a basic foundation of knowledge for our listeners to understand that when we talk about different therapies that are being developed, whether they've already pre-existed and they're being used so-called off-label for this particular virus, or whether they're new medicines that are being created, they're typically either targeting the entry stage or the replication stage of the process, the life cycle of a virus. And that to the extent that we can think about those therapies in those terms, it helps advance our understanding and put things in perspective in terms of the development of these medicines. But the third thing that I find fascinating is that, to me, this is a double-edged sword in a way, from the perspective of what we all want, right, which is a solution to this horrible problem that we're all confronting, is that there's still a tremendous amount of basic science occurring. And on the one hand, that is incredibly reassuring that there are people like yourself, doctor, that are working to better understand the most basic processes and mechanisms of the virus. And those insights may lead to the medicine or the therapy that ultimately gets us out of this predicament. But at the same time, that's a long process. It's probably the case that the medicines that would come from or will come from the basic science that you're engaged in today are not 30 or 60, 90 days away. They're probably quite a bit further out on the horizon than that. So it's great to know the basic science is happening, but it suggests there's still so much we don't know at the same time.
Yeah, you're right. But suppose in 2002 and we hadn't stopped studying SARS, or we hadn't let that lag, had we really pushed on vaccines or antiviral drugs at that time, maybe we would be there now, right? So That's right. Is true that basic science is slow, but it's important, because if you look back to even 40 years ago, if we hadn't done any of that work when SARS first arose, we would have been clueless. We wouldn't have known how to deal with it at all. So I think what you said is absolutely correct, but I also think that it's important to keep plugging on the basic science forward.
Oh, yeah, of course.
Slowly forward.
I mean, we may be lucky and hopefully we will find a vaccine, but that doesn't negate the fact that there are going to be many aspects and facets of how these viruses operate that we don't understand. Just because we find a vaccine doesn't mean that we've unlocked our unlimited knowledge of the vaccine. And so we continue with the basic science regardless, but as we advance the basic science, it really increases the chance over time of coming up with therapies and medicines, because we understand how they operate better. We know what to go after.
Yeah, that's absolutely true. But I mean, I think a vaccine would be really, really wonderful. I mean, that's what's really going to make people feel safe when they go out on the street. Now, I mean, even when we start relaxing, staying home, I think that having a vaccine would make me feel a lot safer.
Yes, me too. So, well, thank you again for joining us today, Dr. Weiss. It's been really absolutely fascinating. And I want to ask our audience to please stay tuned for part 2, where we're going to be joined by Dr. Ofer Levy from Harvard Medical School to discuss advances in therapeutics and vaccine development in terms of specific therapies. Armed with the knowledge that we've taken today from our conversation today with Dr. Weiss, we'll be able to apply that to the conversation with Dr. Levy in a couple of days when our next episode is released.
I want to thank our listeners for joining us, and I encourage you to visit WilmingtonTrust.com for a roundup of our investment and planning content. You can subscribe to Capital Considerations on Apple Podcasts, Spotify, Stitcher, or your favorite podcast channel to ensure you get updates on future episodes. Thank you again for listening.
This podcast is for information purposes only and is not intended as an offer or solicitation for the sale of any financial product or service or recommendation or determination that any investment strategy is suitable for a specific investor. Investors should seek financial advice regarding the suitability of any investment strategy based on the investor's objectives, financial situation, and particular needs. The information on Wilmington Trust's Capital Considerations with Tony Roth has been obtained from sources believed to be reliable, but its accuracy and completeness are not guaranteed. The opinions, estimates, and projections constitute the judgment of Wilmington Trust as of the date of this podcast and are subject to change without notice. Wilmington Trust is not authorized to and does not provide legal or tax advice. Our advice and recommendations provided to you is illustrative only and subject to the opinions and advice of your own attorney, tax advisor, or other professional advisor. Diversification does not ensure a profit or guarantee against a loss. There is no assurance that any investment strategy will be successful. Past performance cannot guarantee future results. Investing involves a risk, and you may incur a profit or a loss. Any reference to company names mentioned in the podcast should not be constructed as investment advice or investment recommendations of those companies. Facts and views presented in this report have not been reviewed by and may not reflect information known to professionals in other business areas of Wilmington Trust or M&T Bank, and may seek to provide financial services to entities referred to in this report. M&T Bank and Wilmington Trust have established information barriers between their various business groups. As a result, M&T Bank and Wilmington Trust do not disclose certain client relationships or compensation received from such entities in their reports. Investment products are not insured by the FDIC or any other governmental agency, are not deposits of or other obligations of, or guaranteed by Wilmington Trust, M&T Bank, or any other bank or entity, and are subject to risk, including a possible loss of the principal amount invested. Wilmington Trust is a registered service mark used in connection with various fiduciary and non-fiduciary services offered by certain subsidiaries of M&T Bank Corporation, including, but not limited to, Manufacturers and Traders Trust Company, M&T Bank, Wilmington Trust Company, WTC, operating in Delaware only, Wilmington Trust N.A., WTNA, Wilmington Trust Investment Advisors, Inc., WTIA, Wilmington Funds Management Corporation, WFMC, and Wilmington Trust Investment Management, LLC, WTIM. Such services include trustee, custodial agency, investment management, and other services. International corporate and institutional services are offered through M&T Bank Corporation's international subsidiaries. Loans, credit cards, retail and business deposits, and other business and personal banking services and products are offered by M&T Bank, member FDIC. 2021 M&T Bank Corporation and its subsidiaries, all rights reserved.